bottom mushroom here. Aha. The bottom mushroom here would be also a hydnum, right? Because it does have a stem, it has smooth spores, and it has, it grows on the ground, looks like it. However, it's actually not closely related to hydnum. The spores are not white, and they don't have stickic basidia. So looks can be a little deceiving, but as a whole, hydnum looks a lot like the mushrooms you see pictured here. So they do grow on the ground in forests, and they are ectomycorrhizal, meaning they are symbionts of common tree species. So oak, pine, spruce, fir, these are some of the tree species that hydnum commonly associate with, and they're found throughout the world in temperate regions of the globe. And as I mentioned, they're usually somewhere on the continuum of white to pale yellow, orange, these colors. They're not darker than a medium dark orange. And as I mentioned, they have these clear, smooth spores, easy to diagnose under a microscope. And these are mushrooms that are all edible. As far as we know, all species of hydnum are edible, and many are commercially harvested and sold in markets around the world. Actually, one of the closest relatives to hydnum are the chanterelle mushrooms in the genus Cantharellus. And this is a relationship that has been borne out by DNA sequencing, but it's actually been suspected for a long time based on that feature of stickic basidia. So stickic basidia is a character that's conserved among hydnum and some of its relatives. But again, very difficult to observe, not something you're going to see under a microscope unless you use highly specialized techniques. So as I mentioned, they are ectomycorrhizal, so are chanterelles. They are found worldwide in temperate areas. You find them in a lot of similar habitats to chanterelles. So if you find chanterelles commonly, you can look for hydnum there a little bit later in the season, like right about now, and you're likely to find them in similar habitat. And similar to chanterelles, they have smooth, clear spores. Other relatives of hydnum now, and Verlé had asked what I'm working on now. I'm not going to really touch too much on it, but I am working on the larger order that contains hydnum. And it's a really interesting order because it contains a wide variety of fungi, not just mushrooms, and fungi that are doing all sorts of different things. They're not all ectomycorrhizal tree symbionts. They are anything from orchid mycorrhizae to lichenized fungi that associate with algae, or fungi that grow on lichens, specialize on growing on lichens, or plant pathogens, such as this Rhizoctonia fungus that is devastating to crops such as potatoes. Crusts on wood that just are saprotrophs that break down dead and decaying organic matter, and even these coral mushrooms in the genus Clavulina. So overall, hydnum is situated within a larger order of fungi that are really fascinating. So just to give you that context. So here in the southeastern US, where I am, I would say that hydnum is a common summer to fall mushroom. We begin seeing them here around June, and they can go through December sometimes. I suspect that your season is a bit shorter as you're further north. However, they are thought as overall a sort of late summer to early autumn. And I think autumn is sort of prime time for hydnum. And as I mentioned, they are pretty common. So I know this will be touched on a little bit more when we talk about the iNaturalist project. But as I mentioned, they are also edible, although I've been told some people find them to be a bit bitter. So there may be a bit of individual variation there. But like chanterelle mushrooms, there are no toxic species. And they've had some cultural importance as they've been featured on many different stamps. Okay, so how many hydnum species do we have? Well, field guides, if you look in them, often have one or two names. And when I began this project, I was finding that field specimens that I was finding here in the southeastern US don't always match the descriptions that were in field guides. And I was obviously interested in eating hydnum, like so many people. But I decided to actually save some of my specimens for science instead. And I began digging into the taxonomic history of Hydnum to understand which species had already been described in North America. So there are two species that were described by a botanist named Charles Peck, who was the state botanist for New York around the late 1800s, early 1900s. And he described two charismatic species of Hydnum, Hydnum albidum, which is small and white, and Hydnum umbilicatum, which is small and orange and is named for this umbilicus or belly button-like feature in the cap, that depression. And I took on loan type specimens of these species. So the holotype, sometimes we are able to get DNA from even old degraded herbarium specimens. These are, over 100 years old, but I wasn't able to get any DNA. So the point of getting DNA from these specimens is to match modern specimens' DNA to the DNA from a type specimen to verify that you have, that you're applying the names to the correct species. Another species that was described from Alabama by Banker is Hydnum albomagnum. This is a much larger white species with really closely packed spines. And this is mostly distinguished by the large size, and it seems to be found with pines. There's often leaf debris that adheres to the top of the cap. And so I also wasn't able to obtain DNA product, but I only found one clade that represented this species in my phylogeny that I was building of DNA sequences. And then there was another species described from Washington, Hydnum washingtonianum. Not a lot of people had been applying this name. And another species described from Maryland, which also this name had never been applied by people after it was described, Hydnum cespitosum. And all I knew of this species was this illustration that was part of the holotype collection. So not a lot of information to go off of. And then there are two European species names that were often applied in North America, and that's Hydnum repandum and Hydnum rufescens. And these are the names that you often see in field guides. Hydnum repandum is the type species, as I mentioned, of this genus, and it's a chunky, cream-colored, larger sized mushroom that has shorter spines that sort of go down the stipe a little bit. And Hydnum rufescens is a medium-sized orange species. So I wanted to know, do these European species occur in Eastern North America? We know with a lot of other mushroom species like the chanterelles that in fact, European species names have long been applied here in North America. However, those species don't actually occur here. There'll be a closely related species that occurs in Europe and then one in North America. So I was compiling a database of sequence data from modern and historical herbarium specimens, and also trying to apply species names. But how do you tell what a species is? Well, traditionally we observe features such as the size, the shape, the color of a mushroom, and microscopic features such as the size and shape of spores, and the presence or absence of other microscopic features. So that's what mycologists have been doing for hundreds of years to describe species. But this approach is a little bit problematic. First of all, Hydnum is not very feature rich. Basically, spore size and the number of spores that are produced on each basidium, that's the spore-bearing cell, are really the two features that we have to go off of microscopically. It doesn't have any other specialized cells like some other groups of mushrooms do. And so it's difficult to figure out what the species differences are microscopically. They may be very vague or almost absent. But DNA sequencing has allowed us to compare the genetic history of different Hydnum specimens and species, and allows us to reconstruct species relationships. And so we do that by taking DNA samples and comparing the DNA sequences of the different samples. So for the Hydnum project, as I mentioned, I was examining and sequencing DNA from Hydnum specimens using fresh collections as well as historical specimens. So if you're not familiar, we use herbaria, sort of like a library of biological collections. And so we have a great herbarium at the University of Tennessee, and these are some photos here you can see. So we have specimens stretching back about 100 or so years, and it's a way to know about species through time. So we have about 70,000 fungal collections in the UT herbarium. So it's a great resource, particularly for the southeastern US, but I was looking at the whole eastern US. Here's an example of a specimen from our herbarium from 1944. So this is a Cortinarius. It's not a Hydnum, but you can see that actually there's a lot of information to be gained from herbarium specimens, even those that are from, you know, so long ago. And so that's just an example of a really well-preserved collection that has a lot of notes from the collector, as well as a specimen that potentially one can gain DNA sequence from. So again, type specimens are representative of what a species is, and that tells us how a species name should be applied. So I was sleuthing into each species name within Hydnum. This is Charles Peck. I mentioned he described 2 Hydnum species, and he was a prolific mycologist who described thousands, about almost 3,000 species of fungi. And here's what one of his type specimens looked like that I attempted to extract and sequence DNA from. And essentially, I followed the process that I described, where I take a piece of tissue sample from the spore-bearing surface, and I sequence it and get a DNA sequence result. And from that, I can compile my list of DNA sequences and use statistical methods to infer a phylogeny. A phylogeny being an evolutionary tree that represents species trajectories through time and shows us species relationships. So I started by compiling a set of not only my DNA sequences from specimens, but also using GenBank, which is a global database of DNA sequences. And I built a global phylogeny of Hydnum. And so I used the DNA barcode for fungi, which is called ITS. I had 398 specimens in my phylogeny, there are 61 clades or species level groupings within this phylogeny of 398 specimens. And I found that 16 species of Hydnum, marked here in red, occur in Eastern North America. So this is in comparison to the previously 5 species names that were applied in North America. And so I'll just go quickly through a couple of things that I learned from this phylogeny. One thing you can see is that all of the Eastern North American species are not closely related. They don't all occur as sister species. So again, a lot of them are closely related to European species. So Hydnum albidum is a species that I mentioned that Charles Peck had described. And here's his protologue or description of that species. Not a lot of information to go off of, just the size, where it occurred, which month. And when I got the holotype of the specimen, it was actually made up of several different collections. So there were actually about 6 or 7 collections representing Hydnum albidum. And in my phylogeny, I found that there were 2 clades that were small white species that I had collected that are pretty common in the southeast up through the northeast. And I had to decide which of these is the true Hydnum albidum. And I started sequencing DNA of the holotype, but I wasn't able to get DNA. So I had to go off microscopic features. But these 2 clades both had the same size and number of spores that were produced. So again, not a lot of morphological differences. And here's Peck's illustration of Hydnum albidum. And so I decided that this species overall fit the stature better, this clade, I mean. And so it was a little bit smaller. And so this one is Hydnum albidum. And this one also had a staining feature that the other one didn't really have. The stains bright orange quite quickly. So, and this one also hadn't been found from New York State, where this species Hydnum albidum had been described. And so in that way, I was able to deduce that this is not Hydnum albidum. This clade is actually an undescribed species, which we went on to name. Okay, so we feel good about Hydnum albidum, and for modern and historical collection records, we know it occurs from Nova Scotia into the southeastern US, plus down into central Mexico, and actually now I know further into South America, in Central and Northern South America. So I would expect that maybe you might encounter this in the woods, although it is quite small, and I've heard that maybe nobody has found it in your area. Anyway, as I mentioned, there were several collections that were associated with Hydnum albidum that Charles Peck had included and called Hydnum albidum. And so even though I couldn't get DNA from the holotype specimen, so this is a holotype specimen here, and these are the other associated specimens, I did actually obtain DNA from 2 of the collections. And I noticed even before sequencing DNA that these had a different spore size than these other collections. And so I knew that they represented a different species. And what was really interesting about getting DNA from those 2 collections is that they matched contemporary collections from Texas, New York, and Honduras. So this is a really interesting species that we named Hydnum vagabundum, the vagabond. And it's much larger than Hydnum albidum. It can grow to about yay big. So this is potentially could occur up into Newfoundland. I'm not sure yet. I don't have any records from Eastern Canada, but since it's up in New York, it's plausible. Perhaps it's up by you as well. And so now I'll go through a couple of other species that are known to occur in like Newfoundland, or that I suspect could occur in Newfoundland based on what we know about these species distributions. So as I mentioned, the things that you want to think about when you find these mushrooms in the field, if you want to identify this, is the overall size and shape of the specimen. You can just measure the stem or the stipe, measure the cap or the pileus, the diameter of the cap, and then the coloration. So some Hydnum species do stain or discolor when you handle them. So that's something you also want to be aware of. What color was it before you touched it? What color was it after you touched it? Did it stain quickly or not? Did it take a while? And then if you have access to a microscope, you want to check out the microscopic characteristics, such as the size and the shape of the spores. And then what we call sterigmata are these little things that stick up on the top of a cell at the edge of the tooth. So under the microscope, you'll see that spores are attached to this basidium by a sterigmata. And so you can count the number of spores that occur on a basidium. So this one has 7 spores. Sometimes that's helpful because the smallest, palest species have the most spores that are produced, whereas, and they're smaller spores as well. Whereas the more orange species tend to have fewer spores on the basidia and they are larger. So these are the types of characteristics that we have for Hydnum. Compared to other fungi, it's not a lot of characteristics, but we'll take what we can get. So Hydnum has been divided into 3 major subgenera, and this is Hydnum subgenus alba. So these are white Hydnum, and as I mentioned, they have the smallest spores, and the most spores, up to 7, are produced on the basidia. And then there's subgenus Hydnum. This is more like your large and chunky kind of classic Hydnum, more white to cream colored, and they can get quite large. They have sort of a medium-sized spore. And subgenus Ruficentia are orange species. Generally, any orange, sort of like darker orange species is definitely in subgenus Ruficentia. And compared to these other species from the other subgenera, they have the largest spores. So in subgenus alba, I'll show you a couple of examples of species that might occur in Newfoundland. As I mentioned, Hydnum albidum, we don't have records that I am aware of from Eastern Canada. However, I would be surprised if it didn't occur in Eastern Canada. I don't know about Newfoundland, but that's a species that is potentially, I don't know, people have told me that they've never seen it. Again, it's quite small, so it's easier to recognize in the field. as opposed to Hydnum albomagnum. This is a species name that people have applied in Eastern Canada, and it's a much larger white species. However, there is some overlap between Hydnum albomagnum and a species like Hydnum subolympicum. So you can see, another thing I should mention is that it's really important to have mature specimens of Hydnum. If they're really young and small and the teeth are not well developed, they may not be producing spores, first of all, and it's difficult to gather an overall size estimate and things like that from the specimen. So sometimes there's a little bit of overlap between a large, really pale species like Hydnum albomagnum and something from subgenus Hydnum like Hydnum subolympicum, because you can see the underside is quite pale, and when these are younger, they might be paler as well. So go for full-size specimens if you see them, and if you have tiny specimens, just keep them for dinner. But this is Hydnum subolympicum. This is what Hydnum repandum is in a field guide to North American Hydnum. This is basically a sister species to Hydnum repandum, that type species of the genus, and it's an excellent edible. I actually found it again the other day here. We don't see it too often here in the Southeast. It occurs often in the mountains, but I know it occurs up. It's actually, the type specimen is from Newfoundland, so it definitely occurs in Eastern Canada, and this can grow quite large. So the specimen I found the other day was jumbo. And so this is a classic subgenus Hydnum. It has spines that generally go down the stipe. We would say they're decurrent. However, looking at some of the specimens that people have brought up on iNaturalist, it'll be interesting to see maybe challenge that assumption. Another species from subgenus Hydnum is Hydnum washingtonianum. So this species was actually redescribed as Hydnum neorepandum recently. However, I found that through DNA sequencing, it's actually synonymous with Hydnum washingtonianum, which I was able to obtain DNA from the holotype, and that's how I was able to match those species. This is also a large, pretty common, I'm assuming, Hydnum that also grows large like Hydnum subolympicum. And so I'm trying to understand how these two vary in the field, and that's part of what will be really helpful about the iNaturalist project that we're working on, is distinguishing Hydnum washingtonianum and subolympicum. But these both occur in your area for sure. And I mentioned Hydnum vagabundum, which stretches all the way from Honduras to New York. And this could very well also occur up in your area. It's large and chunky, similar spore size to subolympicum. I should mention that actually washingtonianum has different size spores, so that is one way to try and distinguish between these species. But this, you can see, is very pale white. It's large, white, and it has this sort of scalloped looking edge. And from the collections we have, they occur in mixed conifer hardwood forests. So again, it's certainly possible that this occurs in your area. And it's also in subgenus Hydnum. And then within subgenus Rufoscentia, I mentioned these are orange species. So we know that Hydnum umbilicatum occurs by you. And this is unfortunately a very highly variable species. So we do see collections where it has this central depression. Often I find that the edge of the pileus has this sort of like whiter edge to it. Maybe it's a little zonate, has these rings around the edge. So here are two examples of verified collections of Hydnum umbilicatum, this one here from New York. However, it can also look more like this. So you can have specimens that don't have that belly button or umbilicus in the middle of the cap. Specimens where the stem is a little bit off center from the cap. So it's highly variable. And furthermore, there are other species that I found that can have that belly button cap, and they're not Hydnum umbilicatum. So these species are really confusing in the field. You also have Hydnum canadense, and this is a species that looks identical to Hydnum umbilicatum in the field. And in fact, the specimen right here, I found at Mount Mitchell, which is the highest point in the eastern US in North Carolina at 6,000 feet elevation. And it was right next to this other Hydnum umbilicatum. And if I didn't use DNA sequencing, I wouldn't have been able to tell them apart. However, you do have Hydnum canadense. And again, this species is probably also highly variable in the field, but is described from Newfoundland. Other orange Hydnum species that I suspect you have in your area, perhaps, are Hydnum quebecense. This is another species that will be very difficult or maybe impossible to tell apart just by examining macroscopic and microscopic features. There is Hydnum submulsicolor. It's really only known from 1 or 2 collections. And however, it very well could be in your area. Again, the spore size should be a little bit different. So perhaps it'd be easier to understand if that's in your area by measuring spores. Hydnum aerostatosporum, I don't have any records from Canada that I know of, but it's very widespread in the eastern US. It can be in mixed woods or under spruce or fir. So I suspect this could also be in your area. So how can we identify Hydnum in the field? Well, as I mentioned, you want to collect fully grown mushrooms. That's really important. And actually with any kind of mushroom, you always want to dig up the entire base of the stem or stipe rather than cutting them off from the ground. Sometimes there are features like staining or whether a stem tapers at the base that are important for identification and good to note in your field notes. And you want to take basic measurements, nothing fancy, just the diameter of the cap, largest and smallest if you have a group like this collection here. And then you want to measure the length and the width of the stipe or the stem as well. And then also note any staining or discoloration, as I mentioned. So this example, this is a species that you probably don't have by you, but this particular species, Hydnum alba aurantiacum, is white at first, and then very quickly, within a few minutes, it stains bright orange all over. So that's an important identifying feature for that species. And so staining and discoloration, you can also cut mushrooms in half and notice if the interior stains or discolors and how quickly. And then taking a good field photo is important. So you can show multiple angles. You don't just want to ever take a photo of just the cap of a mushroom. Take a photo of the underside. You know, if you can include a couple specimens, all the better. You know, pose them at different angles. And not on this list, but make sure you note what the habitat is. So what kind of trees are present? Is there birch or is it just spruce and fir? We find that some Hydnum species, we don't have any clear necessarily understanding of which species of trees Hydnum prefer. We do know that some species like Hydnum umbilicatum are always found with conifer, but they can also be in mixed woods. But it's just important to note, you know, is there pine there or is it just spruce and fir? So just noting the tree species, that sort of thing is quite important since these are actomycorrhizal species that associate with trees. You can use a key to Hydnum species that we have in our paper. This is the paper and it's open access online. You can use that as a sort of provisional key to species in your area. You know, as I do more sequencing of specimens from Newfoundland in particular, but also just along the eastern US, it may become more clear, hopefully, which species you have in your area. But for now, this key is applicable to all of eastern North America. So you can try and key your specimens out in the key in our paper in the meantime. So just a few takeaways. Hydnum are extremely common species found throughout North America as a whole. And there are far more species than we previously thought, right? So in Eastern North America, we thought there were 5 species. It turns out there are at least 16 that we know of so far. And some of these species are really restricted in their range. So we have species that are found or known only so far just from the southeastern US. But then we have species like Hydnum vagabundum, which I mentioned, which ranges from Central America up to New York or perhaps even your area. So really interesting genus. And I look forward to seeing what you all collect in the field. And a few more things, just real quickly. I just want to mention that the species that we have in Eastern North America, only one of the species that I found also occurs in Western North America. And only one other species occurs in Eastern North America and Europe. So we have a really interesting group of Hydnum species that are different than those in other regions of the world. And so again, I just want to press on you that the classic names from Europe, Hydnum repandum, Hydnum rufescens, don't apply to our Hydnum species in Eastern North America. But anyway, thank you for your attention. I'd be happy to answer any questions you may have. And yeah, thanks again for having me.
Great. Thanks very much, Rachel. Verlé, did you have a presentation as well, or did you want to show anything? Oh, you're muted right now.
Uh-oh.
Oh, there we go. Oh.
There we go.
Okay, great.
Okay.
Yeah.
Yeah, thank you. Thank you. Sorry about that. Yeah, I just have, I just want to talk very briefly about the Foray NL iNaturalist project, if that's okay, just for a few minutes. And maybe, like, would you, or would you like to answer questions now, Rachel, about your talk? What would you prefer?
Either way is fine. If people have
Okay.
any burning questions, they could ask now. Perhaps they could just save it until you're done.
Yeah, maybe
Okay.
if folks do have questions now, they can just enter them in the chat, and then when Verlé is finished, we'll go forward with the question. So feel free to type them out, or at least indicate you have a question.
Okay, Levi, maybe you could bring up the Hydnum project on your screen. So I'd like to thank Joanna for the invite, and Rachel for your presentation. Every time I hear it, I learn something new. So I'm just, it's great to hear you hear it again and see some of those new slides. Very exciting. So my name is Verlé Harrow, and as Levi mentioned, I'm on the board of Foray NL and have been on it for the last couple of years. And my husband and I live on Bell Island, Newfoundland. So we're right at the very end of Bell Island, which is in Conception Bay. So it's 2 by 9 miles. And we've been here, I guess, for the last 23 years. And every summer I've been out looking at mushrooms. And for the last 8 years or so, I've been photo documenting the mushrooms on Bell Island. And, you know, eventually I'd like to work up to an iNaturalist project on the fungi of Bell Island. And I just want to say that, you know, even though I have a master's and PhD from MIT in the School of Architecture, I know nothing about biology. So the learning curve has been very steep and ongoing. So, you know, I really thank Rachel and everyone else for their patience, you know, as I sort of, you know, learn about this whole process of identifying mushrooms. So the genesis for the Foray NL Hydnum project was to get our Foray NL community up and running on iNaturalist. And partly this was in response to COVID, but also iNaturalist is an amazing platform for documenting and identifying fungi. And this is particularly, as I mentioned, important because for the last 2 years, we haven't been able to hold our face-to-face forays. The Foray NL's primary focus is contributing to the internationally recognized herbarium at Grenfell. And Rachel has just underscored how important these herbaria are around the world. But if you go to the Foray NL website, you'll see the quarterly publication of Thelina, the species lists for the province, links to Flickr accounts, which have a vast library of photo images of what's collected during the forays, as well as many other fungal related resources. And I just want to say that Foray NL is really quite an astonishing achievement, given that it's a volunteer organization. So it's just mind-boggling how much work has gone into this Newfoundland collection. And of course, this was all started by Andrus Wojcik, who's the author of the Little Book of Mushrooms of Newfoundland. And of course, everybody has that book in their back pocket. When I mentioned to Andrus last spring that we were launching the Foray NL Hydnum project, he became very, very excited because Carl Linneman had sequenced some of his personal Hydnum collections, but it was too late to contribute those to Rachel's work because she was, way down the road on this article that's published on MicroKeys. So the potential of that initiative lay unrealized until the Foray NL Hydnum project came along. And I must admit, it's taken us all summer to get organized, but we now have the iNaturalist site up and running, thanks to Sarah Jenkins. And Levi, you could just type in Foray NL Hydnum project, and that'll take you right to the website. So Sarah Jenkins, who's the editor of Infolina, got the website up and running, and observations are starting to come in. I think we're up to 45 now, and I expect by the end of the season we'll have 50. And this is we're slow to get off the ground, but if you look at the Facebook streams, you'll see a lot of Hydnum are being collected by persons locally. And next summer, we'll be tapping into that and asking them to submit their collections or upload their observations on the iNaturalist site. So this is really, I would say, a 3-year project, and we're just finishing up with, the collection period of year 1, but there's still tons of work to do over the winter. And we're thrilled because Rachel has very graciously agreed to be the iNaturalist lead identifier. So we're totally in her debt. So what are we actually doing and what are our goals? Well, we want to know what Hydnum are here in the Newfoundland, and we want to develop a local key so that when folks are out in the woods, they can identify what they found. And to that end, and after much toing and froing, Andrus has prepared a 3-key approach to identifying the Hydnums of Newfoundland. And we just got this up and running this morning. Sarah got it up and running. So if you go to this site and you look at, I think if you just scroll down, anyway, you can find it. I had a look at it this morning. It has keep going here. There you go. Oh, stop. Yeah, so here are the musings of Andrus Voigt, and he has proposed a 3-key approach to identifying the Hydnums using Rachel and Carr's publications and the iNaturalist project in combination. So key 1 is obviously what we're working on right now, and working from the species descriptions in Rachel's and Carr's publications, we're making a key of species that we're likely to encounter. And if you click on Andrus's, I think highlighted where, yeah, right here. I think that should take you to it. We were having a little problem this morning. Yes, okay. So here's key number 1. And what he's done is outlined, you can keep going down, groups for sure we have based on the sequencing of cars, things we're likely to find, possibly find, and unlikely to find. So that's sort of like step 1. But in addition to that, we need to develop an Excel spreadsheet or a cheat sheet that captures all the information, about the cap, the spines, the stem, the bore, number of basidia, habitat, and distribution. So we need to create an Excel spreadsheet where we can assimilate all these finer points. And then key 2, which again, as Andrus described, is a better tentative key. And we're going to develop that over this year and next year. And we're matching what we found to what we have listed in the Excel sheet. And of course, this is where it gets very woolly very quickly for someone like myself, a citizen scientist, community scientist, very rank amateur. And that is, my observations might might not be capturing the information that we need. And, I have to remind myself that it's a description and it's not creative writing. And so, so we need very clear direction from Rachel, and we'll be working with her to get an absolutely sort of firm list about what we should be collecting, because like there's so much more information that's in the in these scientific descriptions that may or may not be useful. For example, there's a small red mushroom that has a Hydnum that has a very strong floral scent. Like it smells just like chanterelles in many ways. So is that a useful piece of information to be collecting? And I noticed such a difference, being an avid Hydnum eater, that when I cook these, some of them have a very strong manure-like smell, and others have no smell at all. Like there are all these little additional bits and pieces. I don't know if they're useful or not, or if we should be capturing them, if they can become part of the key as time progresses. But maybe, that's something that we'll be sorting out with Rachel along the way. So once we've got our descriptions and we've got our Excel sheet, we are going to be matching the two together. And this is where it would be really great to partner up with some of the MUN biology students. Like I have collected, I don't know, I posted 26, but I probably will have about 35 by the end of the summer. And all of those have been dried. So I really do need someone to get out. My microscopic skills are next to zero. So I really would like to partner with someone who can look at spore size and shape and count the number of stegmata. So that's a really, if someone feels so inclined, by all means, get in touch with me. And next year we will be a little more organized, and there will be some of us who are collecting and drawing. So we'll have more of a collection and need more folks with microscopes. So if you're not able to this summer or fall or winter, maybe you can join the project next year. So that pretty much brings us to key 3. And, of course, as Andres has said all along, oh my God, all we need to do is just have all the collections in the Foray NL herbarium sequenced. Well, that's not going to happen. That's simply not going to happen. So we have to have another approach. And there are going to be, Rachel is going to, as I understand, sequence a representative sample. And so we're very grateful for that. And that'll help us determine, get a better match with the Excel spreadsheet, and also with the micro and macro observations that we'll be making on what we've collected. So in a nutshell, that's pretty much where we are and what we're doing. Andres is always saying, and of course, I think we've discovered all there is, Verlé, so don't get your hopes up. And so, if you were to help us with the microscopy, maybe we could put your name on the on a publication, but we couldn't necessarily name a new species after you. So, there is that limitation to the project. So anyway, that's kind of where we are and where we hope to go. And if anyone would like to be involved, we'd be absolutely thrilled to have you join us. And maybe Rachel has some comments on what we're doing and where we're going, and we look forward to those. And I should have mentioned that Joanna very graciously offered to help with the microscopy. So once she gets completely set up, we'll be pressing her into service, but maybe she can strong arm some others into joining us on this endeavor. Rachel, maybe you'd like to say something about the project overall?
Sure, yeah. I guess my main comment is if anyone decides to participate in iNaturalist, for example, just learning to get a Hydnum 2 subgenus first is really helpful. Microscopy is the really like the next step after that to try and pin down maybe a potential species name or two. But you know, even just making observations, writing out a nice description, size, shape, you know, odor, any of these characteristics is a really great start. And specimens can always be dried, and so microscopy doesn't have to be done right when you know, have a collection that you've made from the field. But yeah.
Great. Thank you very much both for the presentations. Very interesting. I'm sure we're all gonna get out there right away and start collecting mushrooms. So we have a couple questions from folks. First is Joanna, so you can go ahead.
Hey, thanks again for the talk. It was really good. And I'm more than happy to partner up with you, Verlé, if you want to learn some more about how to do the spore sizing. I'd be happy to get together with you to talk about that. I guess one question I just had was Sean Dawson. He's a forager at the St. John's Farmers Market. He often has Hydnum species there, just sitting on his table, ready to eat. I was wondering if that is like a feasible specimen for you. Like, can I pick them up and then size the spores, or do you really need like location, exactly where it's going to be found, and all of those things?
So I would say, because we don't yet know which species are found in your area. I mean, we have some idea, but we don't know all of them. And because different species that are closely related can look really similar and even occur in the same area, like right next to each other, it makes it difficult to make sure that you're not getting like a mixed collection when you're looking, if you take like a couple of specimens, say from his table. And it is really useful to get habitat information at this point. So unless, you know, he could tell you that information, I mean, if you want to try, if you think that, I just think that to start out, it's really good to try and get a sense for what you're seeing in the field first. And then maybe if you see something on his table and you're like, oh, I haven't seen a specimen that look like that yet, you could take, you know, maybe one fruit body. It's just very easy to make a mixed collection is what I'm trying to say from somebody else's specimens.
Yeah, I guess if they're all sitting in the same basket too.
Yeah.
Very helpful. Okay, great.
Yeah. I just want to say that Sean was offered to help with this project, but he doesn't have a cell phone. And, you know, which is just so helpful for capturing that moment. But, you know, it is entirely possible, Joanna, that you could chat with him and ask him to make, you know, 3 distinct collections and, for example, and with, you know, some kind of description, like maybe he could call you from home when he gets home that night or something. I mean, this is a guy who is way, way out in the woods. So he's going to see stuff that we don't, but yeah, it all comes home in one basket. So that's no good.
Okay. Yeah. I can maybe coordinate with him and get him to them.
I could mention a few collecting tips, you know, if you have a basket that you're using. I actually often use like plastic tackle boxes that are large enough for mushrooms. Of course, Hydnum can be quite large, but another thing you could do is like, I use sometimes wax paper just to like wrap up. I kind of like twist off the ends. You know, you can make a real large piece of wax paper to surround like a collection. So even just like two Hydnum mushrooms that are, you know, from the same patch, you know, the largest and the smallest, let's say, you know, he could put those into wax paper, and then that stores really well in the refrigerator. You definitely don't want to store mushrooms in plastic, you know, like a plastic bag or something, but wax paper allows some gas exchange. And, you know, like he could even give those mushrooms to you the next day, and you could take notes on it, you know, measurements and whatnot. And it would probably still be fine. The only thing that you couldn't observe is like staining, right, or color change, but that's one possibility if somebody else is giving you the collection.
Okay, well, he's at the farmer's market tomorrow, so I'll go ask him.
Excellent. I think Yolanda had a question there too.
Yep.
Yeah, Yeah, hi. Great talk, really enjoyed it, and hoping to make some contributions Verlé to the local project. Just haven't managed to have enough time in the woods. It's been a ridiculously busy fall. And also, once we are fully set up in the lab, you're welcome to come use my microscopes anytime as well.
Thrilling.
I'm
Thank you.
not particularly skilled at microscopy either, but one of my PhD students has quite a bit of experience. But my question was more just related to the habitat question and the sort of species association. So, you know, Rachel, you mentioned that you see certain species underneath, you know, or affiliated with certain species of trees, but I was wondering whether the sort of condition of the individual tree and or the stand that tree is in. So if it's, you know, a wet stand, like if it's wetland versus upland, or whether the tree is older or younger, or, you know, more decrepit, does that change the probability of detecting some of those species?
Yeah, that's a great question. And it's something that we don't necessarily know a lot about for many mushroom species. I'll just tell you what I know from my observations of Hydnum. So, for example, Hydnum subolympicum, I know Andres has said that it's found, there always seems to be birch there when he's found it. So that's probably a birch associate in your area. I don't know, I haven't been to those woods, but here I've found it under beech. So perhaps it just prefers hardwoods. And I find it in mature forests. They're not necessarily old growth forests, but just a mature stand of trees. I don't know much about, you know, otherwise the condition of the trees, but generally it's, you know, lesser disturbed areas, I would say. Don't often find them in urban areas here, although it's not impossible. So, you know, I would say that generally for to find more fungal diversity, we do associate a higher fungal diversity with, you know, maybe less disturbed forest types. But other than that, it's hard to say, you know, is there fungal succession, for example, on some of these tree species? Perhaps, but it's kind of an understudied area. So, yeah.
Cool, thanks. Thank you.
All right, do we have any
Rachel,
more?
when you say,
Nope, go ahead.
Rachel, when you say charismatic, is that a scientific term, or that's just how you feel about it?
No, I think
I find it
it's just, I guess I use that in the sense that it, you know, like Hydnum umbilicatum, for example, they're constantly saying, like, when I go to forays here, you know, in the Eastern US, like people are like, oh, this one has, you know, it's Hydnum umbilicatum. It has that umbilicate cap. I'm like, well, that's great. You know, people know and are really familiar with some of these species just because of like their obvious features. So that's what I was hinting at.
Oh, yeah. Yeah. No, people do get very excited about them. I mean, they're a very exciting fungus to come across in the woods. Yeah.
They are. Yeah. The first time I found a Hydnum, I thought it was just a really large chanterelle. And then I turned it over and there were spines rather than, you know, gill folds. So yeah, they are. And they're exciting because you can eat them.
Yes. So do you think do you think there are more species yet to be discovered or not? What do you think?
There may well be. I think in Eastern North America, you know, I think it's definitely possible there could be a few more species. I wouldn't expect there are many more. There are some new species that I've identified with a collaborator of mine from Central America. So, you know, in other regions extending beyond, I particularly sampled from the Eastern US. You know, so up in Canada, there may be perhaps another species or two that we're not aware of yet. But in other parts of the world, yes, there are species yet to be named.
Okay.
Do we have any more questions from folks in the audience today?
Oh, I have one last question. When you say the albinum, when you say it's small, like how small?
So the cap generally doesn't get much larger than this.
Okay.
It's quite small.
Okay.
It can actually be as small as this, even
Okay.
at a mature state, with the spines fully grown. So it's distinctively smaller than the other larger white species. It's definitely
Okay.
diminutive.
Okay. All right. All right. Well, you've inspired me to go back and just look a little harder or try a little harder with the subgenus. I was so nervous just getting the stuff up on iNaturalist.
Sure. Well,
Anyway, I'll go back and try harder.
yeah.
Yeah.
Well, no problem in taking a guess, no matter if the guess is right or wrong, right?
Okay.
All right. Well, if that's everything, then I'd like to thank you both again for joining us today and giving the presentation, and good luck with all your work. This looks like a really cool initiative. Great.
Thanks for having me. Thanks everyone.
Thank you. Great talk.
Okay.
everyone.
Thank you all. Great to see you again, Rachel. Bye-bye, everyone.
Bye.